who is the Associate of Dermatology at University of California, San Francisco, and Director of the Psoriasis and Treatment Center there. Anne will cover atopic dermatitis, and Wilson will cover psoriasis. Thank you.
Thank you, Amy. We're both delighted to be here to give you a compare and contrast between psoriasis and atopic dermatitis. I'm pleased to be joined by my dear friend and colleague, Professor Anne Bocock. So in terms of conflicts of interests, I do serve as a research investigator for these companies, and Dr. Bocock has no conflicts to disclose. So today, we thought we'd highlight the differences between psoriasis and atopic dermatitis as a culinary battle between myself, Iron Chef Wilson. I'll be representing and supporting Team Psoriasis, and I'll be battling my colleague here, Iron Chef Anne, who will be representing and supporting Team Atopic Dermatitis. So may the best chef win. So, as we've just heard, both psoriasis and atopic dermatitis are complex diseases with both genetic and environmental contributions. We'll be focusing right now on the genetic portion, and we'll be comparing and contrasting for you in 3 courses. So for our appetizer round, we'll be discussing the heritability of each of these diseases, as well as letting you know the gene discovery success to date. We'll then move on to the entree round, where we'll each discuss our signature gene for each disease, as well as discuss how these diseases impact important biological functions such as barrier, antigen presentation, and cytokines. And then we'll finish off with the dessert round, discussing the role of ethnicity in influencing the genetics, as well as the role of rare genetic variation. So let's go ahead and start with round 1, the battle of heritability. So by heritability, we're referring to the proportion of disease that can be explained by genetics as opposed to environmental contributions. So in terms of psoriasis, we know that it is quite familial. In terms of twin studies, identical twins are 70% concordant for psoriasis versus only 23% for non-identical twins. If you speak to patients with psoriasis, approximately 1 in 3 will report that they have a family member affected with psoriasis. And the sibling risk ratio, the risk if your sibling has psoriasis, is 4 to 11 times the rate of the general population. Now, there's a heritability number that ranges between 0 and 100, and for psoriasis, that number has been estimated to be about 68%. So, Chef Anne, tell us about heritability for atopic dermatitis.
Right, thank you. So atopic dermatitis, the onset usually occurs within the first 2 years, and as you heard extensively about prevalence, when I read the literature, I could find estimates of 30% of children. It appears that it's a bit less than that, and only 3% of adults. Again, it appears as though it's a bit more than that. But we don't know as much about AD as psoriasis with respect to the lambda S, for example, proportion of first-degree relatives in families. I suspect it's even higher than psoriasis. And of course, with atopic dermatitis, there's this observation that 80% of cases have high serum IgE. In terms of concordance with monozygotic versus dizygotic twins, it's very similar to psoriasis, so 72% versus 23%. And for AD, the heritability is estimated at between 75 and 80%. You did. All right, so for the battle of heritability, the winner is atopic dermatitis.
All right. So you may have won that round, but you haven't won the war. So now we're coming to round 2, the battle of gene discovery. So for which disease have we actually discovered more genetic loci? So what I'm showing here are 9 genetic loci in psoriasis termed psoris 1 through 9 that have been discovered over the past several decades using largely family studies. So this is aggregating large multi-generational families and using a technique called genetic linkage analysis to identify these gene regions. The problem with this approach is that these genetic loci are quite large, and it's very difficult to hone in on the causal genes using this approach. And furthermore, it has been difficult to replicate these different loci between different investigators and different studies. And so more recently, investigators have turned to a different way of thinking about these common diseases, which is sort of the common variant hypothesis. So rather than there being one or two genes that are really driving the disease, we now think of psoriasis and atopic dermatitis as a disease where it's really the cumulative effect of multiple variants that sort of tip this scale and predispose a person to developing disease. And using this sort of new paradigm, a new approach has been devised over the past decade called the genome-wide association study. This is a case control approach whereby you use these DNA chips to measure polymorphisms across the entire genome, up to 1 million different single nucleotide polymorphisms in one experiment, and comparing at each position along the genome differences between cases and controls. And this technique has been wildly successful. So this is a graph showing you the success for psoriasis. Back when I started in about 2008, we knew 3 psoriasis genes, including the HLA locus, IL-12B, etc. The following year, that more than doubled to 8 genes. 2010, 16 genes, and now, as of 2017, we're upwards of 64 psoriasis genetic loci known. So quite impressive in terms of gene discovery efforts based on this new approach to discovering genes.
So in atopic dermatitis, as with psoriasis, the linkage studies with families have given us about 9 regions as well, but as Wilson said, as with psoriasis, these are large regions. It's very hard to find a single gene, and so we've resorted to GWAS, genome-wide studies. And for atopic dermatitis, in 2006, there was 1 gene, which was filaggrin, and I'll tell you a bit more about that. You heard about it earlier as well. 2009, there were only 2, 2013, 11, and as of 2017, there's 34 regions. Actually, it's 30 regions, and then if you do what's called conditional analysis, you get another 4 regions. So I have to say that in the battle of gene discovery, the winner is psoriasis.
Aha, very good. Okay, so let's now move on to our entree rounds, and each of us will be discussing our signature recipe or gene. So for psoriasis, my signature dish is sea bass, namely HLA-C bass, the HLA-C 0602 allele. So just to refresh our biology, what is HLA-C? HLA-C belongs to the MHC class I protein family and is expressed on all nucleated cells, and one of its main functions is to present intracellular antigens, such as viruses, to CD8 positive T cells. So HLA-C 0602 refers to a particular version or allele of HLA-C and has a frequency of about 5 to 10% in the general Caucasian population. And if you look at these genome-wide studies, in every study done, HLA-C 0602 is the most significant gene, whereby a carrier of this particular allele has about a 4-fold increased risk of developing psoriasis compared to a non-carrier. And not only is HLA-C 0602 important genetically, but it also has some very important clinical implications. So carriers of this allele are much more likely to develop psoriasis at a younger age. They're more likely to have affected family members, and they're also more likely to have extensive, more severe skin disease. C0602 is more commonly seen in patients with guttate psoriasis, and are also more likely to have the so-called Kebner phenomenon, whereby minor injury to the skin can provoke the onset of psoriasis. One of the more exciting observations over the past 2 years has been that carriers of C0602 seem to respond more favorably to ustekinumab treatment, or IL-12/23 blockade. So starting now to get into the pharmacogenetics of these genes.
So my signature recipe, or a gene, is filaggrin mignon. So filaggrin is expressed in the granular layer of the skin, late in epidermal differentiation, and it's probably involved in cross-linking of keratins. But it does somehow lead to a disruption of the skin barrier, and loss of function mutations are found in 10% of the European population. And in 42% of atopic dermatitis, you see these loss of function mutations. So the odds ratio conferred by these loss of function mutations is between approximately 3 to 4 or 5. Now, filaggrin, the structure of filaggrin is just shown on this slide, and I don't want to hit Wilson with my laser, but filaggrin is an interesting gene. It has a lot of small repeats, many repeats called filaggrin repeats, as well as a binding site for the S100 calcium EF proteins. And the number of filaggrin repeats can actually vary between individuals, and it's also been shown that the number of repeats can be lower in cases than controls. Now, the two mutations that were first identified were this mutation in the first repeat, arginine 501, which turned to a stop, so it truncated the filaggrin protein to a very short protein, and this deletion of 4 bases at position 2282, which also truncated filaggrin. So you ended up with a very short filaggrin protein in the skin, which presumably had a dominant negative effect, or just didn't do what it was supposed to do. So for the signature recipe, the winner is a tie.
Very good.
Okay. So now we have another battle.
So round 4, we're going to be talking about the battle of the epidermal barrier. Of course, the age-old question has been, is this an outside-in or inside-out disease? In other words, barrier or immune function, which comes first or which is more important? So what's not well known is that actually for psoriasis, there is a barrier gene that is important. So in 2009, deletion of the late cornified envelope 3B and 3C genes was found as a major risk factor for development of psoriasis in a Caucasian population with an odds ratio of 1.38, so about 38% increased risk. And this result was replicated in other ethnicities, Chinese and Mongolians. But interestingly, when psoriasis investigators investigated the role of filaggrin, there was no association of filaggrin with psoriasis. So although both diseases have epidermal components, they seem to be quite distinct. And this is just at the bottom a zoom in of chromosome 1q. You can see there are 5 late cornified envelope genes, and that 30,000 base pair deletion in red encompasses those 2 genes.
So in atopic dermatitis, it goes without saying that defects of epidermal barrier are central, and that clinically barrier repair with emollients is a mainstay of treatment and prevention. And you heard about that as well with infants at early stages of life in preventing AD development later on. The psoriasis LCE3B deletion that lies in the epidermal differentiation complex is not found in atopic dermatitis, so there's no association there. Emma Gutman has done an interesting study looking at gene expression of components of this epidermal differentiation complex and showed there are broad defects of this in AD skin lesions. Filaggrin is also important, though, in leading to other phenotypes in addition to atopic dermatitis, and this is a review from Alan Irvin and Erwin McLean. And it shows that the filaggrin mutation itself will confer a risk of developing atopic dermatitis of about 3.1, but that it also leads to an increased risk of asthma with an odds ratio of 1.5. And then if you have and that's in the presence of atopic dermatitis. If you don't develop atopic dermatitis, you don't develop asthma, which is interesting, suggesting there are systemic factors from the skin. And then there's also an increased risk of food allergy, or in particular peanut allergy, conferred by filaggrin mutations. And there the odds ratio is 5.3. So the barrier is very important and can lead to other diseases. I'm going to talk a little more about GWAS later, but it's also interesting that in genome-wide association studies, a number of the associated genes in these regions can also be associated with asthma or asthma and allergy. So there are genes that are associated with just AD, and one of these is actually a common variant that is close to filaggrin that might regulate its expression, and other components of the barrier in particular. And then there's a couple of genes that are also associated with asthma, IL-13 and CLEC16A, and then there's genes associated with AD, asthma, and allergy, and they're listed here. And those are a number of immune genes, including the MHC. So in the battle of the epidermal barrier, the winner is atopic dermatitis.
Okay, so let's move on to round 5, the battle of the human leukocyte antigen locus, or HLA. So we've already told you about the signature gene in psoriasis, CO602, but what's remarkable is that the signal in the MHC region is so large for psoriasis that even after you account for all the effects of CO602, multiple other HLA alleles pop up as being independently associated with psoriasis, including HLA-B, HLA-A, as well as the class 2 gene DQA1. So really, if you think about psoriasis, it's not a association with just one HLA gene, it's really association with multiple HLA genes. And remember, HLA have multiple functions. Of course, they present antigens to the immune system, but they are also involved in directly regulating innate immunity through NK cell receptors and dendritic cell receptors. So these HLA are quite important in multiple facets for psoriasis.
In atopic dermatitis, there is an association with the MHC that is reported. It's with HLA-DRB1. The odds ratio is 0.89, so it's not nearly as pronounced as in psoriasis. And there's also some evidence for some other HLA alleles conferring similar small elements of risk or being protective. But ultimately, in atopic dermatitis, there isn't nearly the signal for the MHC as there is in psoriasis. And this is a study that I'm doing with collaborators Mark Lathrop and Lauren Mockery, looking at data in the UK Biobank, which will ultimately consist of data from 500,000 individuals. So far, we've been able to look at 150,000 individuals in the bank and look for associations that come up with self-reported psoriasis and self-reported dermatitis. So you can see in these Manhattan plots that consist of negative p-values up the y-axis for evidence for association, and then the locations of SNPs along the chromosomes listed from 1 to 22, and those are and each of those dots is a SNP. And so the red line is the level, the p-value that's considered a significant genome-wide association result. And you can see very strongly that with psoriasis, there's a very, very strong hit with the MHC. It actually goes off the charts. We have to truncate it. And then there's also a strong hit with IL-12B that encodes P40. And then there's other hits, actually novel hits, that we're trying to replicate now in collaboration with Wilson. And then with dermatitis, there's very little association with the, little evidence for association with the MHC on chromosome 6. And then, of course, this very strong hit on chromosome 1, where filaggrin resides. So you can see the big difference in association with psoriasis versus atopic dermatitis. The strongest hit in psoriasis being HLA-C or CW6, and with atopic dermatitis being filaggrin. And in either case, the reciprocal does not apply. So in the battle of HLA, I have to concede to Wilson that psoriasis is the winner.
All right. Okay, so moving on to round 6, the battle of cytokine loci. So of course, we've all been hearing that for psoriasis, a particularly important cytokine is interleukin 17, right? And of course, generation of the Th17 response depends on the cytokine interleukin 23. So what I'm showing here in this diagram, I've highlighted with a red outline all of the genes that have been implicated with psoriasis genetic studies and in relation to this particular pathway. So as you can see, both subunits of the interleukin 23 protein, P40 and P19, are also psoriasis loci, as is the interleukin 23R receptor. And then downstream of that, you have TYK2, JAK2, and the transcription factor STAT3, and each of those three have been also identified as psoriasis genes. So virtually every step along that pathway, from IL-23 to binding its receptor to downstream effects, has been implicated in terms of the genetics of psoriasis. Now, of course, for psoriasis, we know that another important cytokine is tumor necrosis factor alpha. Now, TNF binds its receptor and also activates an important inflammatory transcription factor, NF-kappa B. And what I'm showing here is that there are 6 psoriasis genes, shown in yellow, that either activate or inhibit that NF-kappa B pathway. And so again, there's a concentration of psoriasis genes along these 2 axes, both IL-17 and NF-kappa B.
So in atopic dermatitis, I'm just showing you here the different roles or the different components, the pathways that the GWAS studies have identified. So we have the barrier, as we've talked about, we have environmental sensing. There's a new association with a gene called langerin, which encodes, encoded by CD207. And then tissue response down here, but the major genes are genes of the immune system. And so the ones in red are the cytokines, TSLP, the IL-4, IL-5, IL-13 cluster of cytokines that are encoded by chromosome 5q31, IL-2, IL-21 that lie close together encoded by a region on chromosome 4q, and IL-31. And then there's also a number of other cytokine receptors shown here and other components of that pathway. One of the most interesting and important, of course, is both, well, TSLP, but also the IL-4, IL-13, IL-5, IL-4 cluster, which is thought to be involved in the development of Th2 cells, which you will not see in psoriasis. Interestingly, in psoriasis, and you'll hear about this a bit more, there is an association with the same region. We don't understand what that means, but certainly for the battle of cytokines, we decided that there's a tie.
Okay. So what Anne and I have been describing so far have been sort of individual studies done either in psoriasis or atopic dermatitis and sort of comparing them indirectly. More recently, a single study directly compared the genetics of these two diseases, studying over 2,000 patients with AD, over 4,000 patients with psoriasis, in comparison to 12,000 healthy controls. And the first thing, as you see, are these Manhattan plots. If these two diseases were the same, you would expect identical mirror images of each other, but of course you don't see that. But what the investigators of the study did was quite clever. They took all of the significant loci and categorized them into one of these four categories. So you can imagine a significant gene or hit could be psoriasis specific, psoriasis only. It could be atopic dermatitis specific. It could be shared effect between increasing the risk of both diseases simultaneously, or it could have an opposite effect, increasing the risk of one while decreasing the risk of the other. So where did the results pan out? So for psoriasis, there were 17 loci identified where it increased the risk of psoriasis but had absolutely no effect in atopic dermatitis. And conversely, for atopic dermatitis, there were 5 AD-specific genetic hits. Now, here's where I think is a very interesting result. How many loci do you think were shared between the two diseases, given that they're quite common in the terms of being red, itchy, inflammatory conditions? The result was 0. Not a single genetic locus was identified that both increased the risk of psoriasis and atopic dermatitis simultaneously. And in fact, the investigators found that there were 8 genetic loci that increased the risk of one disease, while at the same time decreasing the risk of the other disease, opposite effects. And that may explain the clinical observation that it is quite uncommon to see a single patient with both conditions. Of course, it does happen, but that's relatively uncommon. And it may be because the genes that are predisposing to one are actually protecting against the other. So that's quite an interesting observation in my mind. And so in conclusion for this study, no loci were identified that increased risk of both, and in fact, there were a number of loci that had opposite effects. Let me move on now to round 8, which is the role of ethnicity. So what do we know about ethnic differences in the genetics of psoriasis versus atopic dermatitis? So for psoriasis, we participated in a study a few years ago, collaborating with our colleagues in Asia, looking at Caucasian versus Chinese psoriasis genes. This was a study of over 19,000 Europeans and 15,000 Chinese. And although there were many genes that were in common between Caucasians and Asians, one thing that came out of this study was that there were actually 10 genes shown in the upper left box that were only specific for the Caucasian population and which were not seen at all within the Chinese population. And this may explain to some degree why psoriasis is more common amongst Caucasians compared to Asians, because there's an opportunity for these 10 genes to be to have hits in these 10 genes, whereas for the Asians, these risk factors do not exist. Interestingly, there's also another type of variation at the interleukin-23 receptor locus called allelic heterogeneity. That refers to the fact that for Europeans, there were 2 independent loci identified at that particular gene, whereas for the Chinese population, there was only 1 independent effect. So even if the same gene affects both populations, there is little subtle differences between those 2 populations. So again, there are distinct ethnic differences. And so Chef Anne, what about for atopic dermatitis?
Right. So what I'm showing you here is a set of genes and the odds ratios for these genes in atopic dermatitis patients from Europeans and non-Europeans. This was a large meta-analysis done recently by Stefan Weidinger and colleagues. First author was Paternoster. So in this slide, you can see here are all the genes. So the odds, this is 1, so this would be if there's no risk. So here we are, the European risk and the non-European risk, or the odds ratios. So the highest, of course, is filaggrin, but again, note that the odds ratios are really small, so less than 1.6, so not even as high as smoking, for example. But then these other genes look quite similar, the risks in both populations. However, there are a couple of genes that have different trends, and this is a novel protein called LINK, a LINK protein, LINK 002299, and interleukin-7 receptor, shown here, asterisk, right at the end, where there's and also STAT3. And that's interesting because STAT3 is also an important gene that's mutated in a rare disease that we'll tell you about in a minute. There's also a couple of loci that could be Asian-specific loci. And this is a CCDC80 gene and an NLRP10 gene, which is a component of the inflammasome, which is quite interesting. And you can see the hit with NLRP10. It's very pronounced in the Asians, and you just don't see it at all in the Europeans. So in terms of ethnicity as well, we also need to look at filaggrin. So these pie charts show the different mutations in Asians versus Europeans on the right. And the spectrum of mutations is really different. So the 2 common mutations in Europeans that I told you about before, you can see one's in mustard, one's in that yellowy green color. But in the Asians, you just hardly see these mutations. And in fact, there's a different mutation, the 3321 del A, and that's the most common gene mutation in Chinese. It's also associated with atopic march, recurrent skin infections, and food intolerance. And in Africa, filaggrin mutations are rare. So there's a great need to look at African populations and what's actually driving atopic dermatitis there, also because the prevalence is so high. So in terms of ethnicity, Wilson concluded it was a tie. Okay.
Okay. So we come now to our final battle, the battle of rare variants. So far, we've been discussing psoriasis and AD as common diseases, but of course, there are rare subtypes that seem to be more Mendelian in nature in that they can be inherited in Mendelian fashion through families. And for psoriasis, this largely is true for pustular psoriasis, so including generalized pustular psoriasis, palmoplantar pustulosis, as well as acrodermatitis continua of Halipo. And over the past 5 years, through exome sequencing or next-generation sequencing approaches, these 3 genes, IL-36 receptor antagonist, CARD14, and the autophagy gene AP1S3, have been identified as having rare mutations that are generally either coding or splice-site mutations that generally drive the disease. And so in a subset of psoriasis cases, there are actually single gene defects that can actually give you the entire phenotype. And interestingly, for CARD14, not only can that give you pustular psoriasis, but it can also drive the onset of familial psoriasis, as discovered by my colleague here, Dr. Bocock.
So in terms of atopic dermatitis, you don't get many rare variants in genes that only cause atopic dermatitis. And so the diseases that I'm showing you here are associated with a number of other features as well as atopic dermatitis. So at the top, we have Netherton syndrome that's due to mutations in SPINK5. Now, SPINK5 actually is associated on its own. Other variants are associated with atopic dermatitis. In terms of hyper IgE syndrome, which is associated with atopic dermatitis, there are variants or mutations in STAT3, DOC8, and TYK2. STAT3 itself is a genome-wide association risk factor as well. So in a way, it's a bit like CARD14 in that it can cause common and rare forms of disease. And then we have Wiskott-Aldrich syndrome, where there are mutations in WASP. There are many other features of, as well as skin in this disease. And then the same goes with IPEX, an X-linked disease, which is due to mutations in FOXP3, which is important for the development of regulatory T cells. And IPEX, of course, is also associated with enteropathy. It can also lead to alopecia, autoimmune diseases against endocrine glands, and a number of other features. So in terms of the battle of rare variants, it's a tie as well. So now we just move on to the summary scorecard.
Okay, so it's been an epic culinary battle between these two heavyweight diseases. So where do we stand? So as you can see, for heritability, atopic dermatitis had the slight edge, whereas for number of gene loci, psoriasis had the clear victory there. There was a tie among the signature gene, HLA-C for psoriasis versus filaggrin for AD. In terms of the barrier, however, atopic dermatitis is king with filaggrin. In terms of HLA, psoriasis had the clear edge with multiple HLA identified versus a relatively weak signal for atopic dermatitis. There was a tie for cytokines with Th17 for psoriasis versus Th2 for AD. There was, in fact, no shared loci found, but in fact multiple opposing loci found in that particular study for number 7. And for ethnicity and rare variants, there was a tie. So overall, unfortunately, we have a draw today, but we look forward to future discoveries and a relitigation of this battle in the future. So we thank you for your attention.
We have time for one question from this simply delicious presentation. Joe.
Joel Gelfand from Penn. Similar to the Oscars, I have questions about the accounting procedures that you used for summing this up. I wonder if you could put a clinical spin on this. How do you counsel an atopic dermatitis patient versus psoriasis patients about what we know about the genetics and how that informs what they should know about their disease?
Yeah, I mean, I certainly, when I'm meeting a psoriasis patient for the first time, I ask about family history, which in age of onset, which gives me a surrogate for what I think is the genetic burden for that individual. And if I feel like they have a higher genetic burden with multiple family members, younger age of onset, more severe disease, that will push me as a clinician to be more aggressive in treatment because they're likely to be more refractory to treatment. But I do counsel them, and patients are worried about potentially transmission to their offspring, and I say that there are many environmental factors important for psoriasis, including temperature, humidity, UV, diet, et cetera. So obesity, smoking, modifiable factors. So I encourage patients that there is a lot they can do to be proactive about their atopic dermatitis or psoriasis.
I mean, I think if you didn't know and you typed for filaggrin and for those rare variants, even those two, and HLA-C, you'd get a pretty good indication of what disease you were dealing with, but not everybody has those variants. That's the trouble. You'd probably be missing 50% of cases.
As the field of pharmacogenomics improves, hopefully we'll have more predictive power that we can develop a gene test to tell who will respond to what medication.
Yeah, I think on the clinical side, at least taking care of mainly psoriasis patients, my patients have one of two concerns. Either no one in their family has psoriasis and they want to understand, well, how is it I have this disease? Or they're concerned that their children are definitely going to have the disease. So I think this information is extremely important and clinically relevant. And the more our clinicians understand how to counsel our patients, I think the better off our patients will be. So thanks.
Thank you, Joel.
Okay, thank you very much.